Culture media methods. Culture media mostly used to culture the microorganisms in laboratory. It's a laboratory technique, and we have a number of microorganisms uh, like fungus, bacteria, virus that simply and rapidly cultivated using these methods. It helps to identify and mm, cultivate the organisms simply. The common methods are cereal dilutions and agar plate methods, ward cup method, slide culture technique, hair bait technique, hair perforation method, germ tube method, single spore culture, and animal inoculations. Firstly, cereal dilutions and agar plate method, most widely used method for the isolation of fungi from soils. Collect the soil samples to be examined. Cereal dilutions and plating the samples, SDA with antibiotics, is poured, incubated at room temperature for 48 to 72 hours. It is the most simple method, most widely used, where the sample should be collected and examined. Then the cereal dilutions from 9 ml, the sample should be at 1 ml. Then the transferring after 7 times, it should be discarded. Then the sample should be taken from either 10 raised to minus 4 or minus 3 and observed under or taken into the petri plate in SDA with antibiotic. It is a BSA medium used to culture the fungus and then incubated to 42 room temperature for 48 to 72 hours. Then we can observe the organisms. Then the second method is walk-up method. It is developed by walk-up in 1915. Easy to perform, less time consuming, and less requirement. Add soil sample in sterilized polythene bag. Take the samples in a sterilized polythene bag and take 5 petri plates at 0.005 to 0.15 gram of air dried soil in each petri plate. Then dispense the soil particles throughout the medium. Incubate the plate at 25 degrees Celsius in inverted position until room temperature. After 3 days, incubate and observe the plate for fungal colonies. It's also another method which is developed by Warkup and a simple method to observe the fungus. Then the third method is slide culture technique used to improve the used to observe the morphological characteristics of mold without disturbing the arrangement of spores and hyphae. Firstly, place a sterilized U-shaped glass rod in the sterilized petri plate. Place a heat-flamed clean slide across the glass rod. Cut a small piece of agar block with the sterilized knife and place the agar block in the center of slide. Inoculate a very small portion of the fungal colony on the four corners of the agar block. Take a cover slips and flame it in. Place gently on the surface of incubator agar blocks. Moist can be provided by the cotton piece dipped in the sterilized water. Cover the petri dish. Incubated at 25 degrees Celsius for upright positions. After 3 to 5 days, the fungal growth is observed morphologically under the low power and high power objectives. Then the fourth method is hair weight technique, used to isolate keratinophilic fungus from soils. Keratinophilic fungus from the soils. Place soil sample in the sterilized petri dish. About 30 pieces of 1 inch soil. Long hair, horse hair is sterilized by autoclaving. 30 pieces of 1 inch long horse hair is sterilized by autoclaving. Spread it on the top of the soils. Wet the surface of the soil with sterilized water. Incubate at 30 degrees Celsius. White to brown fungal growth appear around the hair. Transfer the hair into the surface SDA plate. Fungal colonies grow on the SDA. Fungal can be identified by the LPCB stain. Hair perforation method done to differentiate between dermatophyte, T. mendagrophyte, T. rubrum as well as canis. Hair perforation method done to differentiate between dermatophyte T. mendagrophyte and T. rubrum as well as Microsporum canis and Microsporum equineum. Place a sterile filter paper in the bottom of the sterile plate. Cover the surface of the plate strip with sterile water. Add some sterilized infant hair into the water. Add 5 to 6 drops of 1% yeast extract to the speed. The reaction incubated at 25 degrees Celsius for 4 weeks. Observe the hair by making a wet smear for the presence of conical perforations of hair shaft. Test the positive hair by making a wet smear for the presence of conical perforations and of the hair shaft. Test positive for T. mendagrophyte and T. canis. Germ tube method used for the identification of Candida albicans. Test is a reliable method and performed is less than 3 hours. It's also called a Raynaud broad and phenomena. The culture of Candida species is treated with the mammalian serums and incubated at 37 degrees Celsius for 2 to 4 hours. A drop of special suspension is examined under microscope. Germ tube can be seen as long tube-like projections extending from the yeast cells. Germ tubes are formed within 2 hours of incubation in C. Dublin fossil. Single spore culture. Within the help of microscope, a single spore lying at some distance from its neighbors is focused and a circular cut is made on the agar with a sterile platinum wire or nichrome wire, and the agar is quickly transferred to the petri dish. The second method is the dilution method. A quantity of spore is shaken up in the sterile water and small drop of suspensions are examined for the single spore. Dilution is continued till the drop contains single spore is obtained. Then the drop is transferred into the culture plates. The animal inoculation. For the observation of tissue phase of dimorphic fungus, animals can be inoculated intracerebrally, intravenously, intraperitoneally, or intratesticular with saline suspensions of organisms using syringe or needles. All instruments should be sterilized prior to use. Care should be taken to inject only required amount of suspension. Distinct instrument used for the inoculations. Then the following route of quantitative of saline suspensions of fungi are project for injections into the mice intravenously, intracerebrally, intratesticular, intraperitoneal. First one is intravenously, an inoculum of 0.2 ml of or less injected into the vein of tail of mouse using the tuberculin syringe. Then intracerebrally, an amount of 0.02 ml. Suspensions of fungal culture inoculated. Needle is inserted just above the midpoint of joining ear and out canthus of the eye. Intratesticular and inoculum of 0.02 ml of suspension in tuberculin syringe is injected for a diagnosis of sporotrichosis. Intraperitoneal, a amount of 0.5 ml of suspension is injected through skins and peritoneum but not deep into the abdominal cavity. That's all. It's a very simple topic and only 8 methods are there to cultivate the fungus.